So what happens when things get screwed up? We said that DNA gets mutated all the time. If the DNA is mutated, then the mRNA gets mutated in transcription, and then the protein gets mutated in translation. Turns out that most mutations, as we said, are what we call silent mutations. It will have no effect on the gene whatsoever. However, we can look at mutations that are going to affect large regions and affect many genes, or there could be just a single nucleotide that's impacted. So these silent mutations would be absolutely no effect at all. We have what we call missense mutations, meaning that you're going to code a different amino acid. And we have things known as base substitutions that may change the amino acid that's coded as well. Or you can completely change the amino acid to one of those stop codons. So perhaps you've got codons, you're going along, everything is fine, but you hit a mutation right in the middle and it puts in a stop codon. Well, right there your protein stops. Whatever's left out here as far as your mRNA strand, ribosome doesn't care. It hit a stop codon, it's done, it falls apart. So this protein would never be made properly. It's what we call a nonsense mutation. So in hemoglobin carries oxygen in your red blood cells. If you have a single one, just one, base pair mutation in your hemoglobin, a single base pair mutation, you actually can develop sickle cell anemia. Because that one base pair changes the mRNA, changes the amino acid, the protein doesn't fold properly, it doesn't carry oxygen properly, and you end up with anemia, or the inability to transfer oxygen properly in your body. That's a single, what we call point mutation. We can also have what we call reading frame mutations. And what that means is we're inserting or deleting part of the mRNA, and it results in a change in our amino acid sequence. So we've got a normal at the top, the single base pair mutation, we saw that's a point mutation. Or they have here a nucleotide substitution, it's the same thing. What if I go in and take out a U? If I remove a U, a nucleotide deletion, I still have to read in threes. So everything after is wrong. You see that? As soon as this mutation happens, everything after is wrong. And insertion is the same way. If I stick an extra nucleotide in here, everything after that happens is wrong. So these are all going to form the wrong protein. And if it's a critical protein, of course, things can go horribly wrong for you. These mutations can be spontaneous simply because DNA polymerase screws up. We can also look at mutagens like we talked about with our DNA damage, where you see radiation, UV, smoking, all those sorts of things that would contribute to a lot of these changes in our DNA structure that then change the RNA, that then change the nucleotide, or change the polypeptide or the protein in the end.